There are 7 total steps in DNA experiment. First is sample selection, second is sample homogenization, third is cell lysis, fourth is protein precipitation, DNA precipitation, DNA desalting and washing. You are at the stage to see the DNA by adding alcohol. That will called DNA precipitation step. Just within 2 minutes you will see the DNA of hair, saliva and plant. Clear? This top layer liquid containing DNA and RNA that come after protein precipitation. First 2 steps, sample selection and sample homogenization is theoretical step. Once you know, you can do with anyone. The chemical utilized at the level of cell lysis, first chemical, that is DNA isolation buffer solution. There are 4 chemicals basically. First one is triaxial, second one is EDTA, third is NaCl, fourth is either CTAB or SDS. Okay? CTAB used for fungal DNA, plant DNA, SDS utilized for all kinds of the animal DNA isolation. Clear? Trace maintain pH of the solution. EDTA bind Ca++, Mg++ ions that stop DNA's enzyme activities. Clear? Then go to the protein precipitation solution. All protein precipitation solution containing phenol, chloroform, isoamyl, alcohol. Phenol is too much smelling, so we generally avoid it to use in regular lab, right? So only chloroform isoamyl alcohol is utilized for protein precipitation. The solution you are using in protein precipitation was chloroform isoamyl alcohol, right? After that you will use 100% alcohol. That's called DNA precipitation solution. It precipitates the DNA and you can see DNA is clearly visible in your tube in the form of glassy threads like a structure, right? After the alcohol, if we centrifuge the tube, then your DNA will stick at the bottom. Open the cap and remove all the liquid into the basin. Only in the tube DNA will left. Because it is in alcohol, keep mouth open, alcohol evaporate, DNA dry. Then what happened once the DNA dry? It is ready for the elution storage process. Then you add water or tris-EDTA buffer solution, that is called T10E1 buffer solution, tris 10 millimolar, EDTA 1 millimolar. That will give you storage capacity to DNA and you can store into the deep freezer for whatever year, up to 1000 years you can store it. Right? What much amount of DNA today you isolated from all the sample? That is, I think, a little bit 1000 times you can test the DNA. So the quality is more important in DNA experiment rather than quantity. 1000 sample you will not use even if you do the PhD. Understand? In 100 times, 200 times everything is finished, all research is done, MSc, PhD is pass out. So you need quality DNA extraction. 3 places you can maintain the quality of DNA. One is the grinding procedure, where you grind the material in a low temperature, no sound, no stroke. Second is mixing. Mixing let you the quality separation and precipitation of protein. So removal of maximum protein will depend how well you mix your sample with protein precipitation solution. And after this, DNA precipitation is normally happen at room temperature alcohol, and also chilled alcohol is used to precipitate the DNA. If you read the protocols in different companies, different protocols of different scientists, somebody using or most of the people using chilled alcohol. In laboratory it is possible to get up chilled alcohol, but in here it's not possible. Then you go for the washing experiment, DNA washing. If the liquid is coming, DNA is coming from solution or salts, there is definitely some salt over it, right? To remove it, once again you add alcohol, mix and centrifuge and discard the liquid, and at the bottom save the DNA. That will give you the quality washed DNA. Right? Once it is done, let it dry overnight or minimum 2 to 3 hours. Your all alcohol from your tube is evaporated and you will see dry pellets of DNA is only left. You just add water or DNA storage buffer solution and put into the freezer. That is your final step. To see the DNA, you need to run electrophoresis. In electrophoresis, there are two buffer solution called TAE, TBE buffer solution. TAE developed by trace CDTA and acetic acid. TBE use trace boric acid and trace CDTA and boric acid. That's called TBE buffer solution. Along with that, small quantity of DNA loading dye and ethidium bromide used to stain the DNA. DNA loading dye just help to load the sample, while ethidium will attach to the DNA in gel and it gives illumination once you expose the UV light. In that exposure of UV light, you take all the pictures of DNA. That's all pictures that you seen in coming in DNA fingerprinting, sequencing and all. That's coming from electrophoresis. There are two kinds of electrophoresis. One is called agarose gel electrophoresis. Another is polyacrylamide gel electrophoresis. You might seen eye lenses that is developed through polyacrylamide gel. Okay, so that used to, like this glass, it is clear and very thin layer it is utilized. In that DNA run and DNA sequencing experiment done. Okay, after this 10-minute experiment is left to do the DNA fingerprinting. 5 chemicals and your DNA 2 microliter will be added in one tube and your reaction for DNA fingerprinting is ready. Just put on the PCR and run gel electrophoresis, take the picture of DNA fingerprinting. That will be complete course of MSc Biotechnology up to DNA fingerprinting level. But in one day, you isolated well, your performance is well, supernutrient transfer is well, and better than master class student and graduates in Maharashtra itself and also in country, right? So whatever we doing at a school level, it's awesome and it's better than college campus doing. Many campuses not provide you the single samples to do the experiment. Here you did individually, whatever missing, wrong, something happened, good and bad, that is good experience to learn it. Right? Thank you, everyone.
Thank you.